we're going to talk about DNA XRD, Rosalind Franklin, and the importance of Fourier transforms. So we've been talking previously about basically XRD and analysis and how we could look at this as like a reciprocal space idea. So we're going to talk today about the tragic story of, not tragic, I mean, it is a little bit sad, of Rosalind Franklin and the importance of knowing Fourier analysis. So hopefully people have seen the following image before. So this, not this. What image is this? So this is actually photo 51. Very famous image. It's basically the first XRD picture, XRD diffraction pattern of DNA. And this was taken by Rosalind Franklin. So Rosalind Franklin, this is photo 51, is the DNA fiber from a calf thymus. So when she took this photo, so Rosalind Franklin was an amazing basically chemist or X-ray crystallographer. So she showed this photo, and Watson and Crick, were able to see this photo, and they immediately, because Watson and Crick were experts in Fourier analysis. So as they were trying to, Rosalind Franklin's group and the Watson and Crick group, and then there's another group at the same time, they're all kind of narrowing in on what is the kind of helical structure of DNA, that double helix, which is a horrible drawing right there. But anyways, so Rosalind Franklin actually had this picture, but she didn't have that background in Fourier analysis. But when Watson and Crick saw this, they could clearly see that this was in this reciprocal space, that the key structure of DNA was this kind of double helix structure. So they saw, okay, let me see if I have the next image. They could clearly see, okay, there's a large distance here and here. So large distances in our XRD photo means small distances. So this distance, so large, so it must correspond to a small distance in our real space structure. So remember, this is reciprocal space. This is real space. So large distances in reciprocal space, small distances here. So this could be the A to T or G distance. We also have kind of this, these kind of bands that are offset by this angle. So that could, again, correspond to kind of the pitch. That's what Ross and Crick kind of, theorized. And you can also see kind of these smaller distances here. So these smaller distances, again, can correspond to larger distances in your structure. So specifically distances like between your pitch and kind of these other bands as well. So they saw this, basically ran with it and reconstructed, the real structure of DNA, and the rest is history. So they're credited with it. Now Rosalind Franklin's been given a little bit more credit, but her story is a little bit tragic, or not a little bit tragic. It was tragic because she was not given credit until after her death. She died at age 37 due to ovarian cancer. So again, she's credited now, and luckily her contributions is kind of recognized by scientists. So she's made her impact, but again, no Fourier analysis, you could miss out on fame and a Nobel Prize. Finally, one little just kind of fun example. You could also do reverse Fourier analysis or an inverse Fourier transform. Fourier transform of an image. So you could, at home or in your spare time, because I know everyone has lots of free time nowadays with the exam one coming up, you can look at this photo, download it in Mathematica, do an inverse Fourier transform, and let me know what's the image that you see. Just, you get to do it for extra credit. Just kidding, there's no extra credit in this crash, but I'll give you a little hint that the image is really great. So hopefully that doesn't violate some copyright issues. But anyways, that's all we have for next time. That's all we have for today. Next time, we're going to get into actually the math of our Fourier analysis, and specifically how we could classify time-varying measurements and make continuous or perform continuous Fourier transforms. So I'll see you all next time. Thanks. Have a good one. Bye.